Nice to see so many of you here. I wanted to do a couple of slides to get you warmed up, and then I'll let Ted torture me after that. So, as mentioned, we are a clinical stage UK-based cell therapy business. We are engaged in allogeneic therapy, so very much off-the-shelf cell therapy candidates. We have two products that we're targeting three different indications. It is stroke disability, so it's not acute stroke, but rather chronic consequences of stroke, critical limb ischemia, and we also have another program that is going into the ophthalmic space for retinitis pigmentosa. We also have a very interesting research program on exosomes. Don't think we're going to have time for much of that today, but it's probably watch this space, something very interesting coming in the future here. We have a very good investor support in the UK, so really the top shelf investors in the UK have invested in us. We did a pretty big fundraise, about $40 million in 2013, and also $20 million grants that we got at that time, and the investors you see here, they joined at that time. So we have two different cell lines. One is the CTX cell line. It was derived from a single fetal neural cell in 2002, and it's an immortalized line that we can keep expanding really almost indefinitely. And this cell line we are using for our two vascular indications, stroke and CLI. And from the same cell line, during production of these stem cells, we are able to extract exosomes for our exosomes research program. We also have developed human retinal progenitor cells, which is a more normal clonal stem cell expansion technology. We licensed in the technology from Shepin's Eye Research Institute at Harvard. And what we licensed in was the ability to grow these cells in a very low oxygen culture. We use 3% oxygen when we grow these cells to mimic what's happening in the back of the eye. And we'll come back to that, but we're about to file an IND for that program. Stroke disability. Stroke obviously it's a big issue. Stroke has been an area where there's been a lot of failures in terms of clinical trials. Those failures have been in the acute side of stroke, so the first few hours after stroke. And what we're doing is going for the chronic side, so any time from 2 months to 5 years after stroke has happened, in order to kickstart repair. I think there's no doubt that there is an important market there if we can address this issue. We have data for in phase 1, 11 patients were treated in phase 1. And we don't use any immunosuppressants, and we didn't see any adverse events nor any cell-related adverse events in the phase 1 study. We did look at the different stroke measures, and you have several different stroke measures here. And we looked at efficacy across them, and what we saw was an improvement across all the different stroke measures that we looked at 3 months after the injection of the cells. And this was improved or maintained at 12 months. So that's good news. And we're following these patients to 24 months, and we will publish those data at the Congress on the 19th of April in Glasgow, the Stroke Congress there. So this gave us the impetus to take the CTX cells forward into a phase 2 trial in stroke to get some more data. And we have done that, and we've started a phase 2 trial. And we're going a little bit closer to the stroke episode now, 2 to 4 months, rather than 6 months to 5 years as we had in phase 1, because we think there will be a bit less scarring, so we think that will improve efficacy even more. And we've added another endpoint, which is called ARAT, Action Research Arm Test, which is a functional endpoint which measures the ability of a patient who has a paretic arm, so an arm with absolutely no mobility at the beginning of the trial. And the goal is to get a patient to be able to grab a wooden block from a desk and move it up to a shelf. So that is a very nice, objective, and very meaningful measure if we can meet that. So our goal is to get 2 out of these first 21 patients in the first cohort to make that, and if we do so, we'll continue to 41 patients. And the last few words is on the Retinal Platform. I mentioned the human retinal progenitor cells. So we're trying here to rescue existing photoreceptors to preserve vision for patients with retinitis pigmentosa. We work with UCL at Moorfields in London, and we're also getting funding and support from the Foundation of Fighting Blindness for this program. For the last program, the IND will be going in at the 31st of March, so how many days do I have? Not many. Back at the ranch, this is what everyone's working on right now, and we're looking forward to getting that IND in, and we're very excited about it. So with that, I'll sit down on the torture chair.
No torture, I promise, Olav. Come join me.
Thank you,
so
Ted.
thank you for that overview. Very interesting, and multiple cell therapy products in development. And I think we'll start off with the chronic stroke program. So I guess the first person that jumps into mind is, other than being chronic versus acute, how does your program differ from the one that's being developed at Athersys?
Yeah, so Athersys, we're a big fan because we love to see positive control data for stem cells in stroke. Now there are some important differences. Athersys, they are delivering 1.2 billion cells systemically in order to reduce the immune reaction post-stroke. They deliver them 24 to 48 hours after the stroke, so it's obviously the acute phase, but it gives you a bigger window than TPA. And obviously that window is very, very short, which means only 5% of patients actually get that kind of treatment. So I think it's a good approach. They have 140 patients in a controlled study, and they will actually present this data at the stroke conference in Glasgow on the 19th of April, in the same session where we will present our 2-year data. So we hope they will be successful. If they're successful, it will improve our patient pool, and I think investors will like to see successful control data in this field. So we're crossing our fingers for them. What we do is quite different. We deliver 20 million cells rather than 1.2 billion. We deliver them locally through neurosurgery, and we do it from 2 months post-stroke all the way to 5 years post-stroke. So it's a very, very different time frame. The way we deliver the cells is through stereotactical surgery. So there's a stereotactical frame, and we bore a hole and we deliver these cells right next to where the lesion is for the stroke. It's about a 90-minute procedure, and the patients are in Europe. They have general anesthesia. In the US, it's actually local anesthesia, and it shows you the Americans are tougher. And
that sounds like torture.
Well, actually, with deep brain stimulation, you have to use local anesthesia because you need the feedback. So actually, it's quite well established, and it's routine for a brain surgeon.
Yep, excellent. Now, you showed us the phase 1 data and some of the benefits that you showed. So why, with the phase 2, why move up to 2 to 4 months? And why switch the primary endpoint to ACAT? Is that right? AKAT or
ARAT.
ARAT,
Yeah.
sorry, ARAT. Why make those shifts from phase 1 to phase 2?
There were probably 2 reasons for the shift to go earlier. One was that I don't think it's really data-driven from the phase 1, because I think we didn't have enough patients to be able to say early or late is better. But there was a belief that if you go earlier, there will be less scarring and you'll have a better opportunity to improve and give even better efficacy. So that was one reason. The second reason is that if you go early enough, the patients are still captured in the healthcare system. So they're still in hospital care. It's a kind of a captive audience. So those were the two reasons for going earlier. I think as we go forward, we're probably going to expand that window more. I think that opens up the market. And if we go too early, the patients actually are not convinced that they're not improving. And really, the patients we're recruiting, we make sure that their deficit is stable, that they're not improving. If they're improving, it's not a candidate for us. But sometimes the patients they haven't kind of given up yet, and we kind of need to wait for them to be ready to go through the procedure.
Understood. Now, how does your proposed mechanism of action differ from what's going on at Athersys? Because my understanding is, especially in an acute situation, it's primarily anti-inflammatory and maybe blocking recruitment of T cells and things like that, and macrophages to the site of injury to kind of reduce that inflammation and scarring. What do you think is the mechanism that these CTX cells are playing over time? Are they actually engrafting? What do you think the mechanism is, at least that you've seen through animal work?
We think the mechanism for our cells is very much kickstart of repair. So they kickstart the growth of new neurons and also new blood vessels. And it might have an impact on the immune system as well. So those things really has a long-lasting effect. In animal experiments, sometimes we see the cells grafting and staying a long time, and other times they don't. We don't think engraftment is necessary at all for that repair action. So it's very much a kickstart of repair that will have a long-lasting effect. It's quite amazing that with a one-shot therapy that you can see effects for 3 months that are actually being maintained and improved at 12 months.
Interesting. And then the phase 2 is ongoing. When do you think we'll get data from that study?
We should have the first 21 patients recruited by the middle of this year. And our endpoint is 6 months follow-up. So that means 6 months past that is when we will see the data. So late this year, early next year.
Great, excellent. Now you're also developing these CTX cells in critical limb ischemia, and we should get phase 1 safety data pretty soon. And you are having to do additional safety work because it's a little different patient population here. But what are your plans for phase 2?
The plans for phase 2 is to start by the end of this year, a phase 2 study, about 80 patients, a controlled one-to-one a randomized study. The reason why we have to do a phase 1 is because we're going with a higher dose than in stroke. So in stroke we have 20 million cells, and in CLI we go in the phase 1 we have 3 dose levels, 20, 50 and 80 million. We'll see which one we land on for phase 2. The phase 1 is a pure safety study. We're actually not looking at efficacy. The patients are not sick enough really to look at efficacy in that study. So it's very much box-ticking exercise so we can get the phase 2 running.
Okay, great. Now, also in your introductory remarks, you mentioned the program that you in-licensed regarding human RPCs. So tell us a little bit about this technology and what your clinical development plans are here. And we'll certainly look forward to the IND in the next week or so.
Yeah, no, we're very excited about this program because it's. I know that everything around ophthalmics is very fashionable now. There is a lot of activity there, and what we're doing is different. We're the only ones that are targeting photoreceptors rather than the RP layer, and we're gene independent. There are a lot of different genes at play. Retinitis pigmentosa is an orphan indication, so we're kind of happy that we can address all of them rather than a subset of patients. Our goal in the animal experiments was to show not only that we can. So RP is a disease where you basically get tunnel vision, and it increases over time until it's completely gone. Which must be a very scary thing to face. And our goal was always that we not only could stop reduce the speed of the loss of vision or even halt it, but actually that we could replace lost photoreceptors, actually turn the light back on. and the animal data that we have is giving us evidence that this is possible. So this is why we're quite excited about this program, and that's why that IND is kind of being pushed very hard back at the ranch.
Great. And as you mentioned, you're AIM listed. Where is the cash currently, and how long will that last the company?
We have, at the last public time point, we had $25 million in cash. That lasts us to Q3 next year. So that means well past a lot of data points that we are gathering right now.
Excellent. Well, Olav, thank you very much for joining us, and thanks for that introduction to ReNeuron.
Thank you very much.